All right, I'm going to get this thing started. I think everyone knows me. I'm Adam Feuerstein, and welcome to another of Stats virtual events. We're calling this one the Asco Recap Live. It is sponsored by W2O. So again, thanks for joining us today. Again, my name is Adam Feuerstein. I'm a senior writer here at Stat. And in just a few minutes, I'm going to introduce a wonderful panel of cancer experts, cancer researchers, oncologists who are going to talk about cancer drugs, cancer drug research, and we're primarily going to focus on a bunch of the data that were just presented at the recently completed virtual ASCO meeting. We've got a lot to cover today. A couple of quick housekeeping notes. At the bottom of your screen, you will see a few icons. There's a chat room feature, chat icon feature, and a Q&A feature. The chat feature is something that you can use to chat amongst yourselves if you'd like. Of course, pay attention to what we're saying at the same time. But if you want to ask our panelists or me a question during this event, and I really do encourage questions, so I'd like you to ask as many questions as you want, and we'll get to as many as we can. Use the Q&A button to ask that question. You'll see that. You can click on that. You can type in your question, and then we will see it on our screens. If you see a question that you wanted to ask but's already been typed in, you can also upvote those questions. They'll rise to the top. We'll see those. All right, so thanks again for everyone for being here. Before we get started today, I'm going to welcome Jim Weiss. He is founder and CEO of W2O Group. Take it away, Jim.
Thanks, Adam, for the great work you and the entire Stat team does. You know, it's an honor to partner with you all on this event today. Good morning and good afternoon to everyone. In case you're not familiar with my company, W2O partners with companies across healthcare to provide analytics-driven digital-first marketing and communication solutions for some of the world's most innovative companies. We care deeply and personally about cancer, which is why we sponsored today. Like many of you, I'm a cancer survivor myself, and I've worked with many cancer companies over the past 30 years, telling this story as it evolved from chemotherapy to targeted antibodies, to immunotherapy, to genomics, to cell therapies, and now CRISPR technology. It's safe to say that Adam and his team have heard just about every pitch from me and my team. It's been a memorable ASCO this year, but maybe not for the usual reasons. Put bluntly, it's been a rough week in America during what has been a very rough few months. Our mission at W2O is to make the world a healthier place, and it's times like these that remind us that our well-being is not only threatened by a deadly virus, but by the age-old scourge of racism. But we're not here to talk about that specifically today. I do think today will remind us how great scientists working together can deliver medicine that can enlighten and heal us all. We need that more than ever right now. As Michael Sakara said, the head of leukemia at the Cleveland Clinic, he was quoted in the New York Times recently saying, "Coronavirus infections may be leveling off, but cancer is still going strong." Given the virtual nature of the ASCO meeting and current events, this year's reach was a bit smaller from a communications perspective. We saw just over 50,000 ASCO tweets this year, a decrease of about a third from last year, according to Simpler, which is W2O's media analytics platform. Despite not getting the usual media attention it deserves, I think history will treat this year's ASCO well, though. There was some really good news, maybe some of the best ever. I continue to be amazed by the ongoing advancements in immunotherapy, the benefits of which are getting more powerful and sustained. The early promise and hope of the CAR T therapies is beginning to get more and more real. As a trustee and board member of the Cancer Research Institute, I'm excited to continue this conversation through June during Cancer Immunotherapy Month. One of the things I'm hoping gets discussed today is the impact COVID-19 has had on cancer research and care, and the scientific through line and similarities that might lead to safe and effective treatments for both diseases. I'm also looking forward to an event CRI is hosting with Carl June and others on June 18th, addressing this topic more fully. I'm optimistic that we're working towards not just incremental change, but with a renewed ability to treat the most intractable cancers like multiple myeloma, triple negative breast cancer, bladder cancer, and lung cancer. Back in the 1990s, when Rituxan and Herceptin were the hot new drugs, Sue Hellman, one of the visionaries I got to work with and for at Genentech, predicted that one day we could make cancer a chronic disease. That promise feels more like a reality than ever. Thank you to everyone, including those here today, who plays a role in research, development, and bringing new therapies to the patients who need them. Now I'll turn it back over to Adam and the illustrious panel you've assembled today and hear what was hot at Asco and what we have to look forward to.
Jim, thanks a lot for those remarks, and you know, it's been some dark days, as you noted, in a string of dark days that we've had for the past few months. But I'm also reminded, I think it was either yesterday or the day before, Mark Lewis, who is an oncologist at Intermountain Healthcare, I saw him tweet out that I believe it was one of his patients. He tweeted out that one of his patients who basically went into remission. And he tweeted that, and it's a reminder that even in these dark days, you know, there are good things happening, and particularly when it comes to cancer care, you know, because a lot of things that we will be discussing, patients are being treated. And so let's, with that note, I'm going to introduce our panelists today. I will start with George Dimitri. George is I'm going to read their titles here. George is Senior Vice President for Experimental Therapeutics and Director of the Sarcoma Center at the Dana-Farber Cancer Institute in Boston. George, how are you?
I'm well, Adam. Thanks for the invitation to join today.
Thanks for being here. Next, we have Dr. Grace D. Grace is the Chief of Thoracic Oncology and the Associate Professor of Oncology in the Department of Medicine at Roswell Park Comprehensive Cancer Center. Grace, thanks for being here.
Thanks for the invitation.
And last, we've got Dr. Marcella Moss. Marcella is the Director of Cellular Immunotherapy and an attending physician for hematology and oncology at Massachusetts General Hospital. Marcella, hello.
Hi, Adam. Thanks for having me.
So I thought we would start, like I said, you know, we've all kind of come out of concluding the virtual Asco that concluded on Sunday. There's a lot of data presented, even though it was all presented online, and we were all watching it from our homes instead of being in Chicago. And I thought that we would start with what I thought was kind of probably the most impactful data, a little bit controversial, definitely buzzworthy. If you were on social media, there was a lot of discussion. And this is the Tagrisso study, the Adoro study of Tagrisso in adjuvant lung cancer that was part of the plenary session on Sunday. A lot of discussion about that, a very large benefit in disease-free survival. Maybe I'll start with Grace, and maybe you can give people a thumbnail sketch of kind of what those data were, and then we can kind of get into some of the details and some of the more controversial topics that arose from that data.
Sure. So this trial enrolled patients from with stage 1B to 3A resected non-small cell lung cancer, and randomized them to either, you know, standard care with or without adjuvant chemotherapy, placebo versus Tagrisso for 3 years. And so the disease-free survival signal, which was really astounding, you know, hazard ratio of like 0.17 is, you know, you hardly see that type of hazard ratio in our trials, indicating a significant benefit of administering this therapy, especially for patients with stage 2 and 3 disease. And when they looked at subgroups, whether they got adjuvant chemotherapy versus no adjuvant chemotherapy, the magnitude of benefit is also similarly seen. We are not too surprised with this. We have some hints that this would be what we would see based on the CTONG trial, from China. They did a similar study, mainly enrolling patients with stage 2 and 3 non-small cell lung cancer with EGFR mutated cancers, enrolling them to a first generation TKI. That study, though, did not elicit an overall survival benefit. And so yeah.
Go ahead. I'm sorry. Go ahead.
So that is the key question in this study, given that the interim analysis was unplanned. I think the Data Safety Monitoring Board had a meeting, and they decided based on the efficacy readout so far. The study completed recruitment of patients a year ago. They have about an average of a 2-year follow-up on these patients, and because of the significant signal seen, they decided to unblind and halt the trial.
So you mentioned the hazard ratio of 0.17, which is incredibly strong hazard ratio. So that translates into an 83% reduction in the risk of tumor recurrence. So if you went into the study, they showed the curves. There was a wide separation in the curves. You rarely see a benefit like this in an adjuvant patient population. So a tremendously large reduction in the risk of disease recurrence for patients who have had surgery, have had their tumors resected. I mean, George, let me go to you. Like, how do you, when you see something like that, it seems like when you look at it, like that's an overwhelming benefit for patients. And I know there was a lot of discussion during the plenary and in the discussant about, whether this will be used and what this means. How do you look at that? And again, as Grace said, we don't have a survival benefit yet. So how do we think about those two things?
We don't know how. Oncologists are the only people in the world who think there are different flavors of survival. You can be surviving with cancer, you could be surviving without cancer, but you're surviving. And so the question of what disease-free survival really means, is it a higher quality survival? Or does it really translate into people living longer? When you say there's a survival benefit to a patient, and then you qualify it by saying, well, it's actually just a disease-free survival. They look at you like you're from Mars. Now, let me go back and say, I love these data. These data are exactly what you want to see when you have a terrific drug. Let's be honest, osimertinib is a terrific drug. It pretty much stays off the normal EGF receptor, so people don't get most of those nasty side effects. They get some, but not as many as earlier drugs. So it really does what a great drug was designed to do, right? Go after the mutation, go after it and shut it down, and keep your hands off the normal EGFR so people don't get those really nasty skin problems that you get with non-specific EGF receptor inhibition. So this is kind of what you'd really want for a single target disease. The mutation's driving the cancer, you shut it down, you should get the tumor to stop as long as the drug is on board. So we saw this, the greater we in the sarcoma and GIST world, saw very similar data with adjuvant use of imatinib, known as Gleevec. When you give that to high-risk patients after their surgery, there have been studies, and we saw tremendous control of the disease as long as people continue to take the kinase inhibitor. So here's a study that is showing tremendous, 83% of patients still have their disease stay away as long as they're taking the osimertinib. But what's going to happen when they're at the end of the 3 years? Well, we kind of can predict that from what happened to the people who stopped taking their Gleevec at the end of 3 years. At the end of 3 years, you wait another 6 months and people's tumors wake up. The key question is, do you just keep micrometastatic people on this drug forever? Which is kind of what we've wound up doing in the GIST world. So, and making extrapolations from one kinase inhibitor to another kinase-driven disease, from GIST to non-small cell lung cancer with an EGFR mutation, is always risky, but that raises all sorts of issues about quality of life, but it's what we do in the GIST world. If somebody's 40 years old and they've got a really high-risk GIST, we know it's already micrometastatic, so we'll say, why wait for it to come back? Go on the Gleevec and stay on it, because we know from Brian Drucker's work, people can stay on Gleevec for 20 years and live.
Is there a question, and I mean, this is maybe a question for George or Grace. Is there a question that's raised as whether or not it's, you mentioned this, whether we should just put patients on it and keep them on it, but what about if a patient takes it, or patient relapses and then starts taking it again? I mean, is that part of the question here, is how we should be treating these people, whether you should just be on it forever, or, you know, is there a benefit to going off it, and if you relapse, if you recur, then going on it again?
Correct. So I think those are questions that, and the gist is a little different from lung cancer, obviously. You know, so it plays, you know, these decisions really boil down to biology as well. You know, how dumb the cancer is. You know, how quickly does resistance mechanisms emerge. So, you know, these are questions we don't know, which the ADORA study, hopefully we will be able to answer. You know, after 3 years of being on therapy, how quickly do they relapse? When they do relapse, what's the time frame? Are they resistant? It appears that, you know, based on the CTONG data, retreatment strategy may help. And we actually commonly use that even in stage 4 disease. You know, after you give a treatment-free interval and re-challenging them with TKI, you may induce another response. So a lot of these questions have to also try to answer what's the underlying biology. And then the key question about the overall survival is that, you know, there is cost to take into consideration. Even though toxicity profile is immensely favorable compared to the first generation TKI, you still get some side effects. There's, you know, a 3% risk of ILD, for example, and not to mention financial toxicity as well. And not forgetting that these are patients that can be cured with standard surgery and chemotherapy. So you have a fraction of patients who actually do not need adjuvant.
Let me ask you a question to follow up, because you treat lung cancer patients. You know, Tagrisso is an approved drug. You know, right now it's approved in the metastatic setting. So here the idea here, or the potential here, is to move it earlier stage into the adjuvant setting. It's an approved drug, so obviously you're not waiting necessarily for an FDA, a regulatory approval.
Right.
And it's already a multi-billion-dollar drug for AstraZeneca. And, you know, as you might imagine, analysts and investors are looking at this and saying another sort of more billions of dollars layered on top. You know, I'll ask you, like, what do you, you know, you've come out of Asco, you've seen this data, like, what would you do if a patient fits the profile, or somebody who might be a candidate for Tagrisso in an adjuvant setting? What would you do?
Right. So I have two hats, right? So I have the purist hat from the scientific, you know, perspective. You know, it's not very completely satisfying without the overall survival. And yet you have this physician hat, and you're in front of the patient. You have to take into consideration what their values are. And in fact, these, you know, we quibble about progression-free survival as an endpoint. You know, some people favor it, some people are adamantly against it. And I've come to learn over the years that, you know, many patients actually value this notion of actually not seeing cancer, even though we tell them it doesn't mean it's not there, but just the psychological, you know, support that their cancer is under control plays a large part in their quality of life. And I think some of the key questions from this trial will be, you know, the pattern of recurrence. You know, are you able to really treat them on time, knowing that right now, even with stage 4 metastatic lung cancer, not everybody gets tested for, you know, even EGFR mutation at diagnosis for multiple reasons. And then there's delay in getting test results. So I can imagine when patients do relapse, maybe by the time you get an answer, they may be too far gone to even start therapy. Mm-hmm. So actually starting it in the adjuvant setting may obviate all those problems. So that will be my key question. You know, what's the pattern of relapse? You know, do patients who actually recur, how many of them actually die before even getting exposed to drug? And that might actually drive the overall survival signal. And that's what we see a lot in the medicines that get moved from second line to first line. You know, a lot of the, you know, with pembrolizumab being approved either monotherapy or in combination with chemotherapy in lung cancer across, all PD-L1. I see the value mainly is, you avoid that subset of patients who never get exposed to potentially effective drug. So moving it up front, I can see that an overall survival signal may be achieved, but the devil is in the details.
So let me ask you, we have a question from someone who's watching, someone who has stage 4 lung cancer, and she has a bunch of questions, but one of them is interesting. She asks, does adjuvant osimertinib or adjuvant Tagrisso cure or just delay recurrence? And I wonder, that's obviously a difficult question to answer, but
right.
We look at this trial, we talk about the survival benefit, whether we will ever see the survival benefit.
Right.
I mean, I guess maybe, is that a question that do you think will ever be answered?
It's very difficult. I don't think so. We wish we have these biomarker, biomarkers that can help us determine like a minimal residual disease status, for example, or risk stratification beyond TNM staging to help us select patients and then to answer that type of question. But if you ask me, I think it's more just delaying the inevitable rather than a true cure.
Grace, let me ask you, Adam, can I just ask a question of Grace? That you guys are lucky in non-small cell lung cancer with EGF receptor mutations because those tumors tend to shed a lot of the circulating tumor DNA. You can detect it in a blood test frequently. So you think that would be a useful biomarker? If somebody's finished their surgery, if they're ctDNA negative, maybe let them go. If they're ctDNA positive, seems more clear to me. That's a much higher risk group.
Yes and no. So what we have found out is that, yes, the shedding of this ctDNA is useful for us in actually initial diagnosis. Now we're moving more into liquid biopsy platforms compared to tumor biopsy platform. But it's also subject to sensitivity of the test if you don't have enough tumor burden.
Right.
So I have a lot of patients with evidently stage 4 disease. You can see it. They present with like maybe an isolated brain, some pleural, like fine pleural mets. You don't detect anything in the blood. You go to the tumor, you find the oncogene.
Right.
So I'm still.
The hidden secret is that a negative is non-informative in liquid biopsy, but a positive is very informative, I think.
Correct.
So Marcel, I want to bring you into this conversation because you've been sitting there very patiently. So thank you. And we can get back to Tagrisso osimertinib in a little bit. One of the other things that I was struck by this meeting was, there was a good bit of cell therapy data presented. We saw really kind of the first proof of concept data from Allogene and their off-the-shelf CAR T for B-cell lymphoma. There was TCR data from Adaptimmune, which you discussed at the meeting. And maybe kind of big picture, what you thought about this meeting from the perspective of cell therapy for cancer and how you thought it went.
I mean, I thought this meeting really shows the field maturing and going in the new directions that people have been hungry for a while. Obviously, the liquid tumors with autologous CAR T made a big splash a couple of years ago, FDA approved in 2017. And now there's really been a hunger, I think, for two things. One is off-the-shelf therapeutics, and sorry about the dog.
There's a dog.
So one is off-the-shelf therapeutics, and
before that we're a very dog-friendly Zoom virtual thing here, so dogs are good.
Timing. So off-the-shelf therapeutics is one area that's I think people have been very hungry for, and then the other is signs of efficacy in solid tumor. And what we're seeing that now is that there's multiple abstracts being presented by multiple different with multiple different kinds of drug products and going after both solid tumors, liquid tumors. There were multiple abstracts on BCMA targeted cells for myeloma. And then there was, sort of larger studies of off-the-shelf studies and Adaptimmunes in solid tumor.
So let's let's kind of dive in a little bit on the Allogene, the off-the-shelf CAR T, because again, they're right. We have the established sort of threshold, I guess, of the autologous CAR T products that are out there for those diseases, for lymphoma and leukemias. And here we have an off-the-shelf product, something that could be given on demand. There's no wait for manufacturing, and these are the first data. And so, what did when you saw those data, and it was in a handful of patients, there were some complete responses. There was some obviously there's side effects, toxicity. What did you think of that? What do you think of those data, those early data?
Yeah, so I'd like to see that there was a reasonable number of patients. And so you have to remember with off the shelf, there's two issues that don't really, that come into play that don't really happen with autologous products. So one is that the T cell receptors have to be removed so they don't cause graft versus host disease. And so that's why all these off the shelf products have gene editing involved. And what we saw was one that used TALENs as a technology to remove the TCR. And then the other thing that happens is you have to have the cells avoid rejection. And that's actually, I think, a slightly more complicated thing to get around. And so the way that Allogene uses is they remove CD52, which is a marker that is on all lymphocytes. And then they really, really strongly immunosuppress the host so that the host cannot reject the incoming CAR T cells by giving the host an anti-CD52 monoclonal antibody, which they've designated ALLO-501. The thing is that if you really strongly immunosuppress the host, there's two sort of confounding things that can happen. One is that because anti-CD52 is on all lymphocytes, you get some anti-tumor response probably just from that. In fact, the drug has been tested in these sorts of diseases. And the reason it didn't develop further as alemtuzumab or Campath is that it causes massive immunosuppression. And so there's reactivation of CMV, and there's opportunistic infections and these long-term follow-ups. And so this is all a bit of a background to say that one of the things that was seen, and it's not clear how frequent it was, is that there's CMV reactivation. And so that really shows how immunosuppressed these patients are in order to be able to get the CAR T cell product that's off the shelf.
So the lymphodepleting agent, just to clarify, so that's ALLO-647 is that they use to lymphodeplete.
Oh yeah, I'm sorry, I get the numbers backwards. Yeah.
And I forever forget the numbers. Yeah, so I mean, I know when we talked about this and I wrote about it, you brought up the CMV reinfection. I mean, what do you make of that? Is that something that you'd be watching further on? I think that they seem, there were 2 doses that of 647 that they used in lymphodepletion. They seem to favor the higher dose. I think they felt like they got better results there. What will you be looking for, when we get more data from that product?
I think, so it wasn't clear to me which one that they're definitely going to go forward with, because it didn't seem like there was a difference in terms of anti-tumor response, whether they had the lower dose or the higher dose. But the higher doses were associated with more infections and a little bit of a higher response rate. And so it overall from the CAR T. And so I think I'd be looking for how frequent the CMV reactivation is and whether it causes any disease. I mean, CMV reactivation is something that we can monitor in blood, and there are drugs to treat it. So you can start treating it. It just gets a little bit more complicated. The key though is that you want host immunity to come back at some point, right? So you want to be able to give these patients their CAR T, have their lymphoma go away, but you don't want to immunosuppress someone for the rest of their life. And I don't think that that's going to happen with the dosing regimen that's just as a lymphodepleting aspect of this therapy. But I think it's different to immunosuppress someone for 3 months than it is for a year or for 2 years. In terms of the number of complications that can ensue. So I would be looking for longer term follow-up data and the frequency of opportunistic infections, even beyond CMV.
And durability, of course, is always, in terms of response, is always something that.
Exactly. Yeah, I was talking mainly about the infections, but the response rate was a little bit lower, I think, with this off-the-shelf product, in the 64% overall response rate, and a little bit lower in terms of CR rate than autologous CAR T products. So I think there's going to be a balance between how great is it to have an off-the-shelf product, but not get the durability of response and perhaps some immunosuppression for some significant period of time, enough to cause, potential risk of opportunistic infections, versus all the hassle and cost of an autologous product.
And that raises a good question, and anyone here can chime in. In terms of that, I mean, what is the threshold for an off-the-shelf product in terms of response rates, durability, versus what we see with the autologous CAR T? Like how much efficacy would you be willing to sort of give up for that? I mean, I know that's a difficult question, but like,
Yeah.
is that, I mean, I guess people, I think because people look at it and I think they wonder, you know, does it have to be as good as an autologous CAR T, you know, in terms of response rate, or could it be a little bit lower, just given all the other sort of, you know, the other conveniences that come along with it?
Yeah, and that's a great question. I sort of, I guess I have my own opinions and biases, but I think ultimately the patients and the market will tell. I think, you know, there's the first aspect of regulatory approvals, and I think, you know, things are on their way that I don't see any massive red flags that would result in like, oh, this is not going anywhere. But at the same time, I think, you know, even autologous CAR T is not offered everywhere in the community yet because it's a complicated therapy, because there's so many costs involved, because of the potential toxicities. And so, you know, I'm not sure yet how much more feasible it's going to be to do the allogeneic products in the community. And then I'm not sure, I think on a patient by patient basis, is this going to be, when you're facing a single person in front of you, is it someone who can wait for their autologous product to be manufactured and maybe don't want to take the potential hit in terms of the statistically probable, you know, complete remission rate? Or is it somebody who needs something now and you don't want to wait the 3 weeks that it'll take to get a product back into them?
So we had a question on just getting back to the anti-CD52, the Allo 647 that they use for lymphodepletion. And someone asked, how does anti-CD52 immunosuppression compare with flu-cy regimens for autologous CAR T?
Yeah, so first of all, actually, in this lymphodepletion regimen, they use all three of them, right? So there's flu-cy plus the anti-CD52,
right?
As opposed to autologous where it's just flu-cy. The CD52 in some ways is much more immunosuppressive because it gets rid of T cells and B cells for quite a long time. It can also have infusion reactions associated with it. The Cy/Flu, the Cytoxan is a little bit more myelosuppressive, but those counts usually come back within a week or a little over a week. And the fludarabine is more B cell and T cell immunosuppressive, but the doses that are used, usually patients' cells will come back. They won't necessarily have completely normal immunity right away, but people generally recover, you know, within months.
Mm-hmm. So George, I want to bring you in because I want to talk about the other cell therapy data that came out that I thought was interesting, which was the Adaptimmune, their TCR therapy. And I want to bring you in because you are a sarcoma expert. And there are a lot of their data that they presented at the meeting was in synovial, I'm pronouncing that right, synovial sarcoma.
Correct.
Right. What were your, I know you looked at those data, what were your thoughts about this approach to treating these patients?
I think it's an interesting investigational approach. It's very similar to the NY-ESO-1 technology that Adaptimmune developed and then licensed to Glaxo. So now we've got two things going after a rare disease. Synovial sarcoma happens to have an important, probably the highest percentage of NY-ESO-1, a cancer germline or a cancer testis antigen, but also MAGE-A4. MAGE-A4 is also pretty heavily expressed on a number of synovial sarcomas. And again, that's because the epigenetics are screwed up in that translocation-dependent sarcoma in a very specific way. So it makes sense. It's another, in a sense, targeted therapy for something that a synovial sarcoma cell just can't get out of. And I like that a lot. If you target something that a cell just can't thumb its nose at you and say, I'm going to drop that antigen, I don't need that antigen. These cells can't necessarily drop those. They define that class of cell to a great extent. So then we get into the issue of, is this a serious disease? Yes, synovial sarcoma is kind of biphasic. When it comes back, some people can live with it metastatic for 5-10 years. It's unusual, but it can happen. But most people die within a couple of years at best, even with all the available therapies. So it is a life-threatening malignancy. And I love the idea of going after either MAGE-A4 or NY-ESO-1. So the data here look interestingly similar to what the Adaptimmune technology, now the GSK technology, has shown us, that with the flu-cy chemotherapy, I get nervous about labeling it as lymphodepletion. It's still chemotherapy. And what we simply don't know is what that fludarabine cyclophosphamide chemotherapy does to a mesenchymal malignancy like synovial sarcoma, just for the reasons Marcella says. We understand what it does to the mesenchymal cells we call blood cells. Remember, synovial sarcoma is just a stem cell sarcoma. It has nothing to do with synovium in the knee. You know, you think about synovium between the bones and it makes the knee joints flexible. This has nothing to do with synovium. It's a misnomer. This is a mesenchymal stem cell malignancy. So maybe it's just very sensitive to a mesenchymal stem cell killer like fludarabine to some extent. We simply don't know. I'd love to see those data in, let's say, patients who don't express the HLA-A2 HLA type, because those patients can't get into these trials. The thing about the CAR T technology is you can give it to anybody. The thing about the optimized T cell receptor technology, and I'll let Marcella, she's more the expert, but the thing about that is you have to have a certain HLA type for the optimized T cell receptor to work. If you don't, you're out of luck. So Marcella, maybe you want to comment on that.
Yeah, Marcella, maybe just and sort of briefly as you can, kind of help us understand the difference between CAR T and TCR therapy. I had to learn that myself, and then also the limitations on this TCR therapy in terms of the kind of patient that can be, because there's only a certain type of person or patient who would be eligible for this, right?
Yeah. So CAR T stands obviously for chimeric antigen receptor. So it uses an antibody to bind on the surface of something on the tumor. And so you don't need to have a specific tissue type or HLA type, but the tumor does have to have that antigen. And that's why for lymphomas and for myeloma, it's a little bit, more straightforward than for solid tumors, because we know a lot about what's on the surface and how to target it. With the TCRs, they recognize peptides derived from the inside of the cell. So most of the cancer mutations that we're thinking about are all inside the cell. So it's really limited to be able to use a CAR. But these TCRs will recognize peptides from the inside of the cell, but they get sort of presented, is the terminology, on the HLA type, or the MHC, the major histocompatibility complex. And so in order for a patient to potentially benefit from an engineered T cell receptor, they have to be presenting that peptide in the right MHC. And so there is a lot more involved in the screening process for a TCR-based therapy, because the patient has to have the right MHC, and you can test that in blood. And the tumor also has to have that peptide antigen. And that's where what George was talking about with regard to synovial sarcoma is that these cancer testis antigens make really good antigens for that disease. There are many other lung cancer, bladder cancer, head and neck cancer. There's many other histologies that actually express these cancer testis antigens, but not necessarily in an obligatory and frequent way, the way it is in synovial sarcoma.
So investors tend to get really excited about very, very, very small amounts of data, and Adaptimmune investors got very excited because not only of the data in synovial sarcoma, but because of some data from a second generation TCR therapy that the company is developing. They had some responses in some of the larger solid tumors. Grace said, well, I think lung was one. I want to say colon maybe was another. I kind of forget.
I think it's ovarian and head and neck.
Okay. Yeah. And so, when you see that, something like that, Marcella, like how excited do you get? Or are you like, this is only a single patient, like slow down?
I think it's somewhere in the middle. I think a single patient who has a complete response and it's durable, I get really excited about. Now, these were partial responses, so they weren't complete. So my enthusiasm is a little bit dampened by that. But again, if we're talking about cancer becoming a chronic disease and we're okay with a partial remission as long as it lasts a long time, then I think I'm also excited. I'm enthusiastic about that. I think the fact that it was a total of 4 partial responders across multiple histologies, on the one hand, it does kind of validate the idea of the sort of platform that it doesn't matter what the histology is, what matters is what the peptide and the target expression is, and the HLA type and those sorts of things. That is good, because then it means you could apply one therapeutic product to multiple different cancer types. But 4 is 4. It's not that many, and it's not that many. It was 1 of 2 lung cancer patients who responded. So I do kind of hold a little bit. I do hold my horses a little bit for that. And they also did get pretty high dose lymphodepleting chemotherapy, the Cytoxan and fludarabine. Now, maybe it's a question for Grace, maybe it's a question for offline, but if we were to use that dose of lymphodepleting therapy in a patient with lung cancer, would you expect a partial response? And how long would you expect it to last? Because then we're talking about how much can we attribute to the T cells versus how much to the chemotherapy.
Sure,
Well, Grace, I'll let you chime in if you have any thoughts.
Right. So, I mean, like alkylating agents were like the old drugs that we use for non-small cell lung cancer, and response rates are like 10% at best. So I wouldn't, necessarily expect a partial response from the lymphodepleting regimen used. But my concern always with these cell therapies for solid tumors are the toxicities because of the antigen that was chosen. How often do you see it in your normal tissues? The durability of the response. The lung cancers like to develop these escape mechanisms. They lose antigen, the involved antigen. So it's like whack-a-mole.
Exactly. And that's what I think is the most sort of concerning, is that at least from the CAR T field, we've seen that there's antigen escape so frequently as a mechanism of relapse. And so in solid tumors that also have heterogeneity, unless it's an obligate protein, which may be true for synovial sarcoma, I'm not sure that it's going to be that for the other histologies that were discussed.
So here's a question for all three of you, and I'll let whoever wants to chime in. Just when we think, again, kind of stepping back, big picture, immunotherapy, targeted therapy. We've seen, we've kind of seen both. We're starting to see some intersection of two, combination therapy. What excites you more in immunotherapy, targeted therapy, the combination of those? Where do you sort of see we're going? We had somebody who asked me, one of the questions that came in was like, what are you excited for Asco 2021? So like a year from now, what would you be excited about? And maybe with respect to that, my question.
Oh, shoot, I was just going to say I'm excited to see everyone in person.
We'll all be excited to be in the same building together, exactly. But other than that, like from a data standpoint, with respect to targeted therapy or immunotherapy. Who wants to go first?
Right. So I think, we've seen, in the past, few months, a succession of, FDA approvals of target therapies in lung cancer. Mm-hmm. So, we've been able to identify all these, small subsets of, oncogene-driven cancers and able to develop therapies for them. So I think the cleanest efficacy signals we're seeing is from that approach. Now, durability is the issue, with target therapies, mm-hmm. Which with immunotherapies that has revolutionized our treatments, and the enthusiasm for immunotherapy will not wane. But clearly not all cancers are immunogenic. You have how can we convert these cold tumors to hot? I think Dr. Moss has her work cut out ahead of her.
And George, what are your thoughts on that?
Let Marcella go, but let me give Marcella the last word. It's not an either or. I'm excited about all of this. I'm also really excited about the whole burgeoning field about DNA damage repair inhibitors. I still think that's huge, because that will help us hit a cancer where it can't repair itself. It'll help create new neoantigens to let the immune system work better. And it's also,
George, the PARP inhibitors, is that what we're talking about?
PARP and many, there's like 40 DNA damage repair relevant enzymes. PARP is just the tip of the iceberg. Mm-hmm. And you've got things like, ATR inhibitors coming. You've obviously got a whole bunch of others coming. So I think you're going to see a lot of that information coming through as well. And none of this is single agent stuff. I think it's very hard to cure cancers, even cancers like GIST or EGF receptor mutant lung, with a single agent. It's going to take combinations of drugs, combinations of technologies, and orthogonal approaches. That's what I really like about the engineered T cells. I actually think we probably will eventually make that a commodity and use that as part of one of many orthogonal strategies. But Marcella, you're developing many of these yourself. What do you think?
Yeah, well, it's a little unfair to ask if I'm more excited about targeted therapy or immunotherapy, because I do my career revolves around immunology, and I'm always excited about that. But I think like George said, I think the combinations, it's not an either or, are going to be really exciting. I mean, we're looking more and more into how T cells are responsive to various drugs. Epigenetic modifiers can change T cell behavior. They can also change tumor behavior. You can enhance immunogenicity of tumor by using small molecule drugs. And in some cases, I think you can also make the T cells, because you can genetically modify them, you can enhance them to be either resistant or hypersensitive to a particular drug. And so I think the idea of using cells is so powerful because you can engineer so much into them to make them do what you want. And then you can be enabled to have really interesting combinations. Now, I may be talking about combinations that will be presented in Asco, 2028 or something, but I think we're going to start to see more and more combinations. Like there's this one drug combination that stabilizes the BCMA on the surface of the myeloma cell. And so that you don't have shedding. And so it really enhances the ability of the CAR T cell to kill every last myeloma cell, because they can't get rid of the BCMA on their surface. So it's just one example of ways that I think specific drugs and everything that we've learned about them can be combined with CAR T cells to really get at sort of these durable, complete remissions.
And Marcella and Adam, there have been a lot of questions about that. What do you like better, CAR Ts or BiTEs?
No, I was actually right. Thank you, George, because I was right about to get to that and maybe put Marcella on the spot a little bit. But we have a lot of questions about, talking about the BCMA targeted therapies in multiple myeloma. What you like, we've seen a bunch of the CAR Ts getting to late stage development now. Getting close to the FDA, and we've got the bispecifics there too as well. I mean, we've got ADCs in this world too, and there has been a kind of a debate. It seems like every time, particularly at the ASH meetings, we have this debate about CAR T versus bispecific when it comes to BCMAs. Chime in, give us your thoughts on where this is going.
I don't know if it's my nature. I'm just conflict averse, because what I've done is actually make CAR Ts that secrete bispecific engaging molecules. So it's really both. I think that they both have a lot of power. Now, I think the question you're trying to put me on the spot on is, if I had a BiTE to get BCMA versus a CAR T to BCMA, which one would I choose? I think it's a little bit early to tell. I think definitely BiTEs are a little, it's off the shelf. It's a more traditional drug. It's dosed multiple times. Now they're starting to come up with ways to prolong the half-life, so it doesn't have to be continuous infusions, which I think was a major barrier before. My sense still is that the durability of response from a course of treatment with BiTEs may not be quite what it is with CAR T. And so I think, again, there's probably going to be issues beyond the science that will dictate which one a particular patient is going to get. And it can include things like, how fast is their disease progressing? Are their T cell counts normal? Can they get allogeneic? Can they get autologous cells manufactured? Would it have to be allogeneic? And would they tolerate the immunosuppression? Would they tolerate the lymphodepleting chemotherapy that doesn't need to be given with BiTEs? And so I think it's always helpful for doctors and for patients to have multiple kinds of therapies available to them and to make individualized choices.
And Marcel,
kind of leave it there.
I mean, when I look at engineered T cells, especially autologous, I'm thinking Henry Ford Model Ts technology, because nobody mentioned anything about point of care production, really. The whole idea they have to ship cells away to somewhere, wait a couple of months, maybe you'll get them back, maybe it won't work. I think it's working now for a limited population, right? But just like you say, those practical things are going to be a wave of the future to help us make that technology.
I mean, I would be a huge advocate of point of care production. I think one of the challenges is the regulatory environment that's kind of evolved is that CAR Ts are considered a drug. And so the FDA process for getting an approval for that is completely different than a procedure or a hospital procedure. And there's actually both advantages and disadvantages to it. I've talked to some surgeons, for example, who do research and they devise new medical procedures. And one of their challenges is that there's never the rigor of the studies to figure out whether their procedure is better than the previous procedure. Not the way that we do in oncology and like what the bread and butter of Asco is. So I think there's advantages and disadvantages to both, but I think in order to make CAR-T more feasible across multiple institutions and communities, I think point of care manufacturing really needs to come into play.
So Grace, we're probably going to done an entire event on lung cancer because there's so much going on.
Right.
So I want to ask you about two things that were, one thing that was kind of big at the meeting was the new checkpoint inhibitor target TIGIT, which I know you discussed in your presentation, the Roche Genentech compound, and then KRAS as another, because lung cancer is kind of playing heavily in the KRAS G12C mutation. Give me your thoughts on those two targets and where you see it going.
Right. So I'm, for disclosure, I'm an investigator in the Amgen 510 study. So I've personally treated patients on the trial. So I've seen what it can do. Lots of patients who were previously progressing on standard therapies or having toxicity from standard therapies, and then you get this mutation-specific drug. We were concerned about potential toxicities and nothing. It's like waiting for toxicity to happen, very negligible, and you see these responses. Of course, it's it doesn't occur in all patients, but 40% of objective response is I guess we have low bar in lung cancer. Works pretty well for a target that had like no therapy against it for decades. So we do see the value. The key questions are again the resistance, both primary and acquired, and so the field is going into what are the combinations to work. So I'm enthusiastic about the G12C pipeline of drugs that are being developed. So.
And Adam, let me kick in. I'm full disclosure, I'm on the scientific advisory board of Mirati, but I love the Amgen drug too. I think the more the merrier.
Right.
So one important thing is we've gotten spoiled in oncology, right? 40% objective responses in lung cancers, but then everybody knocks it and says, but it doesn't work in colon cancer. What good is it? It's like, come on. So we need combinations. We know we're going to need that. And
Right.
colon cancer is a very different malignancy
Correct.
from lung cancer. So
Right.
I'm impressed. And I actually think, it's funny how flip we are that you remember how big news this was when Amgen first presented those data with, I don't know, what was it, Grace, 20 patients? And it was like
Right. 12 patients.
So it was a year ago, right? It was ASCO
Right.
2019 where we got the first data in lung.
Right.
Right.
Right. So again, it's a start of a technology. It's a start of really going after it. Look at how many years RAS avoided being drugged, and now
Right.
we've start.
Right.
So getting to, do you think, it's not necessarily a provocative question, but ASCO 2021, will we be talking about the first KRAS approved, KRAS targeted drug approved?
I'm hopeful. Yes. I'm hopeful. Yeah.
And then
The data is, I think, from a clinician standpoint, very convincing. I mean, vemurafenib was approved despite a short, duration of, progression-free survival, the initial, course in melanoma. So I don't see why not in lung cancer.
And we have just a few more minutes, so I'll go back to you again, Grace. TIGIT or TIGIT, however we pronounce it, hard G, soft
Right.
G.
Right.
What's your thoughts about that as a
Yeah,
target?
so it's this holy grail. What combination immunotherapy can have increased efficacy with minimal toxicity, and at least preclinically, this approach may be viable, and that's actually what was seen in the randomized phase 2 study. The toxicities are quite favorable, but I'm not sure what to make of the efficacy signal, partly or largely because the control group did so much poorer than what you would expect. So you wonder about confounding imbalances in some unmeasured way. We are not aware of what the underlying, for example, molecular phenotype. And if you look at the 95% confidence intervals for the PFS, it pretty much spans what you would expect from, monotherapy with the other agents. So I think the jury's still out there. I mean, it's still a first step. You have a positive trial. It deserves further testing, but, phase 2 to positive phase 3, 60% success rate.
So follow-on question, and I know this gets deep in the weeds, but I'll ask it anyway because someone asked here. Regarding TIGIT again, do you think an active FC is relevant or not?
It depends on your point of view. So if your contention is that you have a lot of Tregs, and so the IgG1 approach to induce ADC to clear out your Tregs may make sense. On the other hand, the concern is, well, you might actually deplete your effector T cells. So I don't know what the answer is, maybe George or Marcella.
I think the FC always matters.
Right.
For a soluble antibody. And it may be similar to like a CTLA-4 situation.
Correct.
So finally, let's end on this. We all went through that. We were watching virtual ASCO. We were experiencing this meeting at home. It seems like ESMO, the next kind of big solid tumor meeting, will be virtual. ASH just announced, I think this week, that it will be virtual as well in December. How do you guys feel about the virtual medical meeting versus, other than the social aspect of not being around and kind of getting that? I mean, how do you guys feel about virtual medical meetings?
I actually didn't mind it too much. On demand, I can do it anytime. I can attend multiple things that I want and not feel like I missed something major. But as you pointed out, the interaction is probably, much to be desired. You don't get to talk or ask questions as much as you would like.
Marcella and Grace, you both had presentations that you recorded for ASCO. Was
Right.
it weird? Was it weird watching yourself speak? I don't know if you did. Maybe you didn't even watch it, but did you?
I hid myself to the edge so that I could just see the chat going, because they asked us to be on so we could answer questions. I found it, in some ways, it was easier to present virtually, but really harder, at least for me, to absorb information from ASCO in this format. I'm not quite sure why. It was a little bit harder to be in the moment, even though I could pause, even though I could look, stare at the slides longer. But at the same time, I think cancer goes on and research has to go on. So I think it's important that that all of the societies are moving forward and presenting this virtually. It's much, much, much better than not doing it at all.
And I love the fact that we could download the slides right away. I think that's really
Right.
important. Everybody can get a front row seat.
Mm-hmm.
I also just miss the ability to go up to somebody later and talk to them about their data. Science is inherently a collaborative sport. So we do it now. We've gotten used to communicating this way, but we do miss it. And I worry the societies are going to worry that this is going to be the future, right? And because they get a lot to support the mission of ASCO, the mission of AACR, the mission of ASH from these big meetings, international meetings. My European colleagues, on the other hand, said, this was great because when people tape things, generally they spoke more clearly and slowly and distinctly. So they found it easier to understand when English wasn't their first language, which is something I hadn't thought about.
Well, with that, I'm going to wrap it up. You guys are awesome, and I really appreciate you spending the time with us today to talk about all of this. I thought the comments were great, and we can't hear people applauding, but I'm asking everybody in the virtual world, please applaud our guests George, Marcella, and Grace for their time today. And I will give you guys an applause.
My co-speaker.
Thank you for having me.
Yeah, but I also want to thank our sponsor W2O for sponsoring this event. And as you guys know, STAT is having lots and lots of events virtually these days. So please stay tuned for more events like these. And with that, we will wrap it up and say goodbye to you guys, and thanks again for your time. Thanks, and
Bye.
thanks everybody for watching.
Thank you. Bye-bye.
Bye.